In this brief presentation, we will describe how BigDAWG has been applied to the ocean metagenomics dataset collected by the Chisholm Lab. This video is a part of the Intel Science and Technology Center for Big Data and is in collaboration with a number of institutions shown at the bottom. This is a little bit of background on the ocean genomic dataset. This data is collected by the Chisholm Lab at MIT, which has been collecting seawater samples from across the globe for many years. In our discussions with the researchers, we have found that they face a number of challenges. First, the time and cost of these expeditions can be quite expensive. The volume and variety of data make it difficult to integrate, explore, or summarize, and the genomic dataset, which is a large part of the overall dataset that we have, contains mixtures of microorganisms, and often extracting sequences to known or unknown organisms can be pretty difficult. Further, correlating metadata with sequence data often results in one-off solutions that don't really expand to other questions or analyses. Just a description of the dataset we're using. It's currently about 12 terabytes, and this dataset continues to grow as more data is sequenced. The data was collected from a number of cruises, and a subset of the cruises that are shown in the bottom left. The different colors indicate different cruises, and each dot indicates a station where data was stopped and data was collected using a device such as the one on the right. And what happens at each one of these stations is that a device such as the one on the right is lowered into the water, and the different bottles that you see on that device actually collect water at different depths. The water is then taken back and analyzed by a number of people for different parameters. The dataset that we have contains 5 different entities. The first is what we call the raw sequence data. And essentially, water samples collected from the sea are frozen, shipped back to MIT, and sequenced at some time in the future. And you're sequencing really to determine what organisms or microorganisms are present in the water. Another piece of the data collected from the same samples, or from many of the same samples, are called the sensor metadata and discrete sample metadata. The discrete sample metadata is a recording of nearly 500 different entities, such as ocean chemistry. This could include information about different metals, about oxygen that are present in the water. The sensor metadata contains pretty deep information about the sensor itself that went into the water, about the recordings, the quality of the recordings, and where they took place. A lot of these cruises are often multi-month, and there are long reports written by individuals aboard these cruises. And these are often free-form text reports that describe the environment or other parameters that might have hampered data collection during the cruise. Finally, we're also using a dataset collected by the seaflow system, which is developed by the Armbruster Lab at the University of Washington, to simulate streaming data or data that might be collected from sensors on board a ship to help better navigate and improve the quality of the cruises people are taking. In order to help address some of the challenges I mentioned earlier, we're developing a number of applications. The first application, exploration, is really designed to help people look at the entire data set and understand what's going on very quickly and intuitively. Navigation is to help cruises become more efficient. So you might be able to optimize your cruise route based on prior knowledge of what type of water or what type of organisms you're looking for. The geoanalytics piece helps researchers leverage the unstructured data and potentially look for gaps in data collection that might be described in these text reports. The genomic processing extracts genomic sequences and helps researchers have a quick look at the full data set very intuitively, and then look at how that correlates with some of the metadata. Some of the heavier analytics, such as that developed by Macrobase and Tupelware, help researchers actually start to look for automated analyses of the data to extract potential insight that they might be able to dive deeper into. And finally, since we're database people, we also have some performance modeling, which describes how well the systems, and in particular the one we use for genomic processing, performs. The BigDAWG architecture contains a number of different layers. The bottommost layer, we're leveraging a number of different engines which are best suited for the particular types of data. So the hydrographic metadata is stored in Postgres, and in fact in different Postgres instances on different databases on different nodes. The seaflow streaming data is ingested using SStore. Accumulo is used for the ship logs. MiriaX and SciDB are used for some of the genomic data, and the different processing leverages the different underlying engines. We've developed a number of islands, as well as a number of different applications at the top that leverage these different islands to talk to different engines via the middleware. The colors over here indicate what screen or what application is making use of which islands and which underlying engines. So it should be noted that this is as a part of the Intel Science and Technology Center for Big Data. A lot of the work presented in this demonstration has really been a joint effort from a number of people across a number of institutions. This is the Explorer part of the Ocean Metagenomics demonstration using BigDAWG. In this screen, we've utilized a number of technologies such as BigDAWG, Postgres, and the visualization engine, Vega. This screen provides researchers a quick way to take a look at the overall data set. As I mentioned earlier, data is collected from a number of cruises, which are given by, and data is collected at different stations, which are given by the red dots over here. What a user can do is quickly hover over these different data points. You can look for a station ID, the total number of genomic sequences that are collected, and the total number of water samples that are often at different depth that are collected at each one of these stations. You can kind of look at the different points over here. Now that you may want to do a little bit more than that, and in fact you might want to look at what are some of the elements in the water or the metal content of the water, the chemistry of the water, and over here what you can do is click through and look for that. So by clicking on phosphate over here, I get a little plot that shows up on the right side, which is essentially a plot of the nutrient concentration versus the depth in meters. So you'll see in this particular example, we had 48 total samples. So you should see approximately 48 different dots over here. Some of these will be at the same depth because you might collect multiple water samples at the same depth. So this is just a quick, easy way for a researcher to look at what's going on with nutrients as the depth changes, and also to understand what data and how many data elements have been, or data points have been collected at any particular station. So you can also add additional nutrients to compare how they may look as you go through. So as at the next point of the cruise, you see a slight change in the nutrients, but you see the overall trend is maintained pretty well. You might see a much different trend if you were to click further away in your cruise. So this is at the beginning of a cruise, this is much later in the cruise. So you see changes, whereas much more gradual changes step to step. So this Explorer screen provides researchers a quick, intuitive way to take a look at what data they've collected, and also to understand any trends that they might see by looking at depth versus nutrient concentration. In the second screen of our Ocean Metagenomics dataset, we show you what it might look like to stream data from ships as they are cruising through the ocean. This screen utilizes a number of technologies: BigDAWG, SStore, the streaming database, and Postgres, the relational database. What we see over here in this red line is the track that our ship has taken so far. This data is being streamed to us. Although it's historical data, we're streaming it at the rate of 1 sample per second. So it might look like the ship is moving pretty fast. We've actually sped up the ship movement just to make the demonstration a little bit more interesting. If you want to look at what we're streaming in, you can kind of look by pressing the button at the top here. You'll see that we're collecting the speed, temperature of the water, a measurement of the salinity, and we're also collecting some information about the different microorganisms that are in the water. And this data is provided by what's called a seaflow sensor, which is developed by the Armbruster Lab at the University of Washington. So we might look at the different organisms in the water, such as Prochlorococcus, Cyanococcus, Piculicurites, or beads, which are just different microorganisms that are in the water, collected and measured by the seaflow data system. So now if we go back over here, we see that our ship is moving along the coast of Hawaii, and as I mentioned earlier, one of the challenges of these cruises can be the expense in just spending these multi-day and often multi-month cruises in the water. And very often you're kind of going around in the ocean not really knowing where good data samples, let's say that are designed to find certain microorganisms, might be. So what we wanted to do was integrate data from the Argo ocean data system, which are a series of sensors spread across the ocean that collect and record different types of information about the ocean chemistry, for example, temperature and salinity. So if I'm a ship captain and I know that my cruise would like to move in the direction of higher temperature, I'd like to be able to real-time steer my ship in that direction. So what you can do over here is select a higher temperature, lower salinity, any combination of those. So in this case, we would want to steer our ship towards higher temperature, and that's given by the green arrow. So that would basically tell our ship captain to take a left turn at the point that they're in, and that you'd be able to see greater temperature. Of course, though, since we're using historical tracks, our ship's just going to move in the direction that we had sort of already collected. You can kind of change that. You can look at if you want to move in the direction of greater salinity, lesser salinity, or any combination of this. So what essentially is happening is we've taken data from the Argo system, which we've integrated with the track that the ship is taking, and we're using simple vector math to estimate which direction would be the best direction to move in, to move. Now, another thing you might want to do, because there's obviously a lot of cruises that are happening in the water, is look at some of the historical data tracks that have happened. So this data, what we're doing over here, is we're using data that's currently stored in Postgres and doing a spatial lookup of what's happening close to our particular cruise of instance. You might use that to look for other historical data, such as, okay, well, let's say I did take a left. What was actually collected the last time? And how long has it been since we last collected data? Another interesting aspect of this, you might notice, is that the color of this line, which was reddish, has now changed to purple. And by looking at the historical tracks, we've actually real-time migrated the data that we've collected in SStore into Postgres, such that so that somebody who might be at another cruise location could quickly look up what this cruise is doing and what its path is. So this screen is just a way for researchers who are on board, who might have sensors collecting real-time streaming information, to get a better sense of where their ship is headed and what they might be able to do to improve data collection as they progress through this ship. The next screen of the ocean metagenomic demonstration using BigDAWG is to help researchers leverage the number of free-form text reports and logs that are written by researchers on board these large cruises. So we're using Apache Accumulo, the key value store, BigDAWG, D4M, and Postgres, the relational database. The first piece, you might just click through the different stations at which data was collected across these different cruises to determine what the station IDs are, bottle depths, and this is using the relational island in BigDAWG to do these queries. Another thing that you might want to do is help explain what some of the gaps across, why are there large gaps across this, why is it more dense in certain areas. So as an example, you might look for, well, there's a pretty large gap in this particular cruise. I wonder if there was a storm, what happened in that cruise. So you might search for the word storm, and this is issuing a BigDAWG query to Apache Accumulo to look for the word storm, and is bringing in data from Postgres to tell us which cruise that this report came from. So we can click on this point over here, and we see that actually the word storm was mentioned 3 times in this particular report. And this pulls up the raw report that the researchers had written. So we see over here that there was a storm that lasted about 4 days that made work impossible, and this is east of Newfoundland. So if we go back to our little map over here, that kind of explains why that there was a large gap in this particular cruise as people were making their way down the Atlantic Ocean. The third screen of our demonstration does genomic data processing and utilizes a number of technologies such as BigDAWG, Myria, Postgres, and Spark for processing. What we have over here is a quick way to pick a number of genomic sequences. So as you know, the genomic sequences that are collected from different samples are correlated with the different metadata that's also collected on board these different cruises. So what you might do over here is select different row depths of interest. So you might want to only look for the sequences or samples related to collections when your depth was under 20 meters. You can update this over here, and what this does is pull in data. It'll only pull in the sample data to those related with samples under the depth of 20 meters. First thing you might want to do is just look for how different samples and depth, for example, correlate with each other. So there are a number of statistics that have been collected based off of the genomic sequences. One such statistic is what we call entropy, which shows the spread of different genomic sequences. So you might look at depth versus entropy, shows you that at about 10, about similar entropy, whereas at about this higher depth, you see greater entropy within your within your things. You might also look for other parameters that you can select over here and look for time series plots over here. At the bottom, where we're doing some of the heavier genomic analysis, we were interested in looking at what parameters or what variables are correlated with how similar or dissimilar different genomic sequences are with each other, or different samples are with each other. So over here at the top, we have different sample IDs, and we see the same sample IDs on the right. And what we're showing over here is what's called the Bray-Curtis dissimilarity, which is a measure of how dissimilar two genomic samples are with each other. So recall that these genomic samples can be many million characters long, and we want to look for how dissimilar sequences are across two different samples. So we'll notice, first of all, that very light means that they're very similar, and as we would probably expect, we have zero dissimilarity between the same samples, so they're the exact same. Darker implies that they're more dissimilar with each other. So we may want to investigate or understand what makes or what parameters might be involved with making two things dissimilar with each other. So that's what you can do over here. We can actually select a particular variable and we can sort based on that. So we see by looking at this particular concentration of this element, there's really not much, at least at a top level, that you can notice. But you might look at entropy, and you notice that if you were to sort based on that the sample IDs have changed order, but you will notice that a lower entropy sample is far more distant from all other samples than any of the other samples. And in fact, as your entropy increases, your dissimilarity with other samples decreases. This might be an interesting aspect for some of the researchers to look at and investigate further. And you might want to then take entropy, for example, and look at how does entropy vary with depth, or how does entropy vary with other data that's collected in these water samples. So this is the first step of our genomic data processing part of the ocean metagenomics demonstration. This screen describes some of the heavier analytics that one may want to perform once we have the genomic data and a lot of the different metadata that we've collected from different water samples. This screen utilizes BigDAWG for the middleware, Macrobase as the engine to look for outliers, Postgres, which is storing a lot of the data, and SciDB, which did some of the pre-processing on the genomic data and migrated to Postgres. You can connect to a number of databases. In order to speed up the demonstration, we've created a sample table in Postgres, which we can query. We could, of course, replace this query over here with any valid Postgres query. First thing you can do is look for different explanatory attributes by selecting plus signs for these attributes. We may also want to look for what types of target metrics we're looking for. So we might want to look for example, we might want to look for where the concentration is increased or decreased. We can go back to the top, and what Macrobase does is look for a number of outliers. So we see over here that for 22 records, the value of these two Levin-Murr sequences was normal. As if we scroll down, for 2 records, the value of this K-Murr, which corresponds to a high CTD-PRS, which is essentially a pressure collected by the CTD sensor, is low. So that's something that you may want to go back and investigate further. This screen, we're still working on working with the researchers to look at what particular metrics are of interest and not of interest. The second part of our heavy analytics screen develops what's called a correlation and spatial model for the data we've been collecting. This screen consists of 3 pieces. The first piece is just a map that shows the cruise, and you might recall this cruise as very similar to the one we took on the streaming screen. You can zoom in to this. What's being collected, and we're using data collected from the seaflow sensor at the Armbrust lab, and this is historical data that we're looking at. Kind of zoom into different aspects. One of the components collected by the seaflow sensor is concentrations of different microorganisms in the water. So sort of zooming into the map, you can see that concentrations are heavier for some parts and about constant for other parts. So the leg of this particular leg of the trip is about constant, but around this where you start to turn around, you see a heavier concentration of the different microorganisms. What microorganisms are in the water? That's given by this correlation matrix on the right side. So the correlation matrix is showing the correlation between Prochlorococcus, Cyanococcus, or the other microorganisms that I mentioned in the streaming demo screen. You'll notice that, of course, each organism is highly correlated with itself, so a perfect correlation of 1. And for the most part, different microorganisms are positively correlated with each other, except for beads, which has a slight negative correlation with Prochlorococcus. Just an interesting aspect. Something else you might want to do is look for how these concentrations varied as you took your trip. So you'll notice over here we have the latitude, longitude versus the concentration in that axis. You can actually move around this screen over here, and you see that one leg of the trip had a higher concentration of Prochlorococcus than the other leg of the trip. You can change that to another microorganism, Cyanococcus, and here it's just not as clear because the Cyanococcus concentrations are just much lower than Prochlorococcus for this particular part of the trip. Another thing you might want to do, because we mentioned that a lot of these trips can be very expensive and you've only traversed certain parts of the water, what you might want to do is look for what the estimated concentrations would be at another point in the water. So we have a spatial model that we're designing over here that's given by this bottommost piece, in which you can enter a different latitude and longitude, and we will estimate what the concentration, and this number is in 1 million cells per liter of water. So this says that there are 258 million cells of Prochlorococcus per liter of water at a different latitude and longitude. And you can play around with that. And this uses a simple linear regressive model in order to do that. So you can look at Cyanococcus, and that's a much smaller concentration. So as an overview, this screen utilizes BigDAWG, Postgres for the historical data, and Tupelware to perform some of this machine learning aspects of it to help researchers quickly understand what's going on in the data set, how different organisms are related with each other, and to estimate what might be going on in different parts of the ocean that your cruise is either unable to visit or has not been able to visit.